All right, welcome to The Explainer. Today, we're going to crack a microbiological mystery that's more than 140 years old. But get this, it's still being solved every single day in labs all over the world.
So, let's start with a problem. I want you to imagine it's the 19th century. You're a scientist. You know the bacteria are causing all these terrible diseases, right? But when you look through a microscope, they're all just these tiny, colorless, almost identical little specks. So how on earth do you tell the good guys from the bad guys? Or even one bad guy from another?
Well, that brings us to our main character, a Danish physician named Hans Christian Gram. And here's the thing. Back in 1884, he wasn't even trying to solve that huge problem. Nope, his goal was way more specific. He was just trying to figure out a way to make bacteria stand out from the lung tissue of patients who had died from pneumonia.
So he's in his lab, just tinkering around with different dyes. He sloshes on a violet dye, then an iodine solution to kind of set it, and then he washes it all with alcohol. And what he saw next was totally unexpected. Some of the bacteria, the ones that cause pneumonia, they held on to that deep violet color for dear life. But others, like the ones that cause typhoid, the color just washed right off. He had accidentally stumbled onto something huge.
And that little lab accident? It turned out to be one of the most important discoveries in the history of bacteriology. It created the very first major dividing line, like a police lineup, for identifying these invisible suspects.
And here they are, the two gangs he basically identified. You've got your gram-positives, which hold onto that deep purple stain, and then you have the gram-negatives, which end up staining a bright, vibrant pink. Just like that, you've got two distinct groups sorted by nothing more than color.
So you're probably wondering, how do today's lab detectives actually do this? How do they get these colors to pop? Well, the beautiful thing is, the process is still this elegant, super effective, four-step dance.
Okay, let's walk through it. Step 1, you flood the whole sample with a purple dye called crystal violet. Everybody turns purple. Step 2, you add something called a mordant, grams iodine, which basically locks that purple dye in. Now, step 3 is the most important part. You wash everything with a decolorizer, usually alcohol. And then for the grand finale, step 4, you add a pink counterstain called safranin. Okay, check this out, because this table shows you exactly where the magic happens. After the first two steps, right, the crystal violet and the iodine, everything's purple, no difference. But then comes that decolorizer, and bam, look what happens. The gram-positive cells, they stay purple, but the gram-negative cells, they become totally colorless. And that right there, that's the whole trick. Now they're a blank slate, ready to soak up that final pink stain. So this leads to the big question, the million-dollar question, right? Why? Why do some bacteria cling to that purple dye while others just let it all wash away? Well, the secret, it's not what they do, it's what they're made of. It's all about their armor, their cell wall.
And really, the entire case comes down to this one key molecule, a thing called peptidoglycan. You can think of it as this strong, mesh-like layer in the cell wall. And the most important thing to know about it, its thickness. The thickness is everything.
And this is where our two suspects are totally different. The gram-positive cell wall, it's got this super thick layer of peptidoglycan. Seriously, it's like 90% of its wall. The best way to think of it is like a big, thick wool sweater that just soaks everything up. Now, the gram-negative cell, it has a really thin peptidoglycan layer. And, this is key, it's covered by an outer membrane. Think of that like a thin t-shirt protected by a waterproof raincoat. So, let's go back to our process. When you hit that thick wool sweater of the gram-positive cell with alcohol, it actually shrinks the fibers. The pores in that peptidoglycan mesh get smaller. And that physically traps that big purple dye molecule inside. It's not going anywhere. It's totally locked in.
But for the gram-negative cell, that same alcohol does the complete opposite. It dissolves its outer raincoat, that lipid membrane. Poof, it's gone. And now, the purple dye that was sitting in that thin t-shirt underneath, it just washes right out. The wall is basically leaky now. And what are you left with? A colorless gram-negative cell, a totally blank canvas. So that final pink stain, the safranin, just swoops in and colors it bright pink. And there you have it. Case closed. Two clearly identified culprits, the purple positives and the pink negatives.
Okay, so this technique is ancient by medical standards, right? Over 100 years old. So why are we still talking about it? Why does it matter so much today? And, like any good detective story, what are the tricky parts? What are its limitations?
It matters because it is, and I'm quoting the American Society for Microbiology here, fundamental. It's still one of the fastest, cheapest, and most important first steps you can take to identify a bacterial infection. Just knowing purple or pink can help a doctor make a really educated guess about which antibiotic to use, and that can save critical hours or even days while waiting for more detailed tests.
But here's the catch. It's not foolproof. It actually takes a lot of skill to get it right. That decolorizing step is super critical. If you do it for too long, you can accidentally wash the purple out of a gram-positive and make it look pink. Don't do it long enough, and a gram-negative might stay purple. Even how old the bacteria are can mess things up. It's an amazing tool for sure, but you have to know what you're doing.
And yet, for more than 140 years, from that one happy accident in Hans Christian Graham's lab to the most advanced medical centers today, this simple, color-coded test has been an absolute cornerstone of microbiology. It's just an amazing testament to the power of scientific curiosity and a little bit of luck. When you think about it, a simple choice between purple and pink can literally be the difference between life and death for a patient. It really makes you wonder, doesn't it? What other simple, game-changing discoveries are out there, just hiding in plain sight, waiting for someone to have their own happy accident?